So this is the last leg of the lecture where I'll be talking about some of the application in water and healthcare as well. So first part is in the water quality monitoring. So if you look at the interest that we had in terms of water contaminants is in the waterborne pathogens. So in water there can be a lot of contaminants. There can be metal contaminants, organic, inorganic. There are huge amount of contaminants, but one of the deadly ones is this E.coli, which is often referred to as the kind of indicator organism. So in other words, E.coli can only enter into water, potable water, is through fecal contamination. So if your water contains E.coli, means that water must be compromised. The water source has been compromised. So the chances of having other deadly things like Giardia, rotaviruses, and so on are extremely high. So that's why we target. This is our target organism, where you can see that some time back I saw that in one of the daily newspaper in West Bengal, talking about there is some E. coli they found, or coliform they found in Ganges. Not surprising, but also in in North America, particularly in U.S. Counties, there are a lot of counties where often there are water boiling advisories. So you don't see or hear them in the newspaper, but typically you will have them close to around 100 to 200 water boiling advisory every year in a lot of the counties in U.S. as well. And also we got hit badly in some time back in 2004, where we had the E. coli outbreak in Canada as well, because one of the water treatments, so where the water gets treatment in a municipality water, it was in Walkerton, it got compromised, the treatment unit failed, and then there was an E. coli contamination and so forth. So it is a kind of not only a problem of local scale but a global scale. So before we get into the understanding of E. coli in details, or rather I want to, before we do monitoring of E. coli, I want to give you some idea of why, how one can look at the cell, the E. coli itself, so that you have a better understanding. So the cell in general, it is a multi-scale problem, because cell is a multi-scale problem, because you have various kind of processes that are taking place. There could be cell adhesion, there could be migration, there could be mitosis. So this is the breaking of the cell nucleus into identical set of chromosomes, cell to cell interaction. So all these are at various scales. Okay. So if I look at in a biological tissue, so this is generic, nothing to do with E. coli, any kind of biological tissue, if I look at the biological tissue itself, what you will see, this is more in the macroscope. You will have all the various cells which are in some sort of a hanging and within a collagen fiber. So these are all the cells. So we'll have a lot of fibers around which is binding this. Okay. So the blue ones are the cells. So this is a typical cell, and this is the The red one is the collagen fiber. Okay. Now, if I focus to one of these cells, so if I look at the cellular scale, if I want to see what is happening, this is what I see in a cellular scale. Okay. So, in a cellular scale, you have a big cell. So, this is you are looking at a cell scale or in the micro scale. So, you have this is the circle. You have a big cell and maybe a portion of a neighboring cell. So, this is a neighboring cell. This is the cell. So, it will typically have a nucleus. Then it will have certain elements which I am drawing here. So, there are some adhesion molecules. There will be some filament. They will end up, I will label each of these soon. So, let me, what is that? So, let me label these. So, this wiggly kind of thing which is around the cell wall, which is these are adhesion molecule. The black straight lines, these are actin filament. You will still have some collagen fibers, and then these dot ones are the focal adhesion point. Typically, that is where they get anchored. You will have myosin motors, which are the red circular ones. And then the blue dots are the actin monomer. Good example will be a G-actin molecule. So, this is typically how the cell, or in the case where there are multiple cell, you will have the structure similar of this nature. Okay, so these are the, so a cell will interact with other cell through these mechanism. They can get attached to this focal adhesion point. You will have individual motor, myosin motor. They may be bound, they may be unbound, various classes of this, and you will have the entire thing. You will have this collagen matrix all around this cell. So these are like from a biological scale all the way to the cell scale. So in a biological cell, you will not too worried about in the bigger scale all these finer details, but when you are in the cellular level, you have to understand all the finer details what is happening in a organisms which has multiple cells. However, E. coli is a single cell organism, which makes it quite interesting. So it's we can actually quantify certain things about the E. coli being a single cell organism. Okay, is it everyone is done with it? Is it? It's still working. So what is a biological term used for single cell organisms? Yeah. So what kind of cell? There are two different primary. There are two prokaryotic, right? So prokaryotic is the one which is for single cell, and the eukaryotic is the one which is multicellular organisms. Okay. So E. coli is a prokaryotic or a cell which is a single cell. Okay. Is it good? So if you look at the E. coli, we can characterize the E. coli by various means. This is a karyotic cell, which is a single cell. Okay. Its cell volume is close to 1 micrometer cube. Cell mass one picogram. Surface area So we can look at how many protein typically E. coli will have. We can calculate that based on the presence of a number of amino acids that are present. So average protein of 300 amino acid each of these amino acids each of them has a mass of close to 100 Dalton. Okay. So the mean mass of protein is close to 30,000 Dalton. 1 Dalton, if you look at the conversion, 1 Dalton is equal to 1.6 times 10 to the power of minus 24 gram. Okay. So, typical protein mass, so if I convert that, so that comes around 5 times 10 to the power of minus 20 gram. Okay. So, then I can say the number of protein in E. coli cell will be the total mass of the protein over the mass per protein. Okay. So, total protein mass is your comes out to be 15 times minus 14 gram over 5 times 10 to the power of minus 20 gram. So, that leaves us around 3 times 10 to the power of 6. So, this is a rough calculation that so much amount of protein that one can expect in a E. coli. So, there are some thumb rule. There are something called a membrane protein as well as cytoplasmic protein. So, we can divide this. So, typically this number that we have can be, as a thumb rule, 1/3 of that is related to membrane protein. Okay. So, we can say that the membrane protein is close to 10 to the power of 6, and cytoplasmic protein two times 10 to the power of 6. Okay. So, one can do a similar calculation for the ribosomes as well. So, ribosomes are the where the synthesis proteins, right. So, this is a very critical component of the cell. So, we can similarly we can do calculation for the number of ribosomes within the cell as well. We can look at the lipid molecules. Everyone is done with this? You have in your undergrad somebody you have done this, or this was not been taught? Microbiology at all has not been? Huh? Little bit. Someone says yes, someone says no. Huh? In grade 11, 12. Okay. Not okay. So, if you look at the lipids, so I am skipping the calculation for the ribosomes. You can do a similar calculation for ribosomes as well. The number of lipid molecules they will be with both the inner layer as well as the outer layer of the membrane. So, they will have both in the inner and the outer layer, with inner and outer layer. So, inner membrane and outer membrane. So, the number of lipids molecule that we see is roughly 4 because you have inner and outer for each of the bilayers. Okay, so you have 0.5, half the surface is covered by membrane protein, area of the E. coli cell divided by area of the lipid. So the 4 is coming because you have inner and outer membranes for each bilayer. Okay, so the lipid bilayer, for each of this bilayer you have 2, so 2 times 2 lipid is a bilayer, so it has 2 layers, so that's why you get the 4. 0.5 is because half of this surface are covered by membrane protein. By membrane protein. Okay. So if you E. coli the lipid, typically the area of a lipid will be somewhere around 0.5 nanometer square. So if you plug these area of the E. coli already provided, so it will come out to be close to 2 times 10 to the power of 7. Okay. So what is in a cell like in E. coli? What is the major constituent of a cell? A cell contains what? It's a fluid, right? So it will have the water as the main component, right? So roughly 70% of your cellular mass by volume is water. Okay? So therefore, mass of a E. coli cell, if you look at it, is 1 picogram. So we can say that it is roughly total mass of water. Total mass of water is around 0.7 picogram. Okay. So then we can say the number of water molecules in a E. coli cell will be the mass, which is the 0.7 times 10 to the power of minus 12 gram divided by the molecular weight 18 gram per mole times what do we have to do in order to get the total number? So Avogadro number, right? So we have to 6.6 times 10 to the power 23 molecules per mole. Okay. So if you do that, then we get somewhere around 2 times 10 to the power of 10 water molecules. Okay. So an E. coli cell will have 2 times 10 to the 10. Okay. Also, it will contain some ions. So like potassium, sodium, so positive charge ions will be there as well. So we can say that typical ion concentration, typically say potassium ion is around 100 millimolar. So the number of ions we can similarly compute (100 times 10 to the power of minus 3), which is in the mole, times the Avogadro number divided by 10 to the power of 15 micrometer cube. So this is 1 liter times 1 volume, which is 1 meter cube. So, this gives us somewhere around 6 times 10 to the power of 7 ions that we have. So, we can actually draw how a E. coli cell will look like. So, if you look at the E. coli cell, so if I draw the E. coli cell, so typically they have flagellas. So, typically at the front they will have these flagellas which help it to move or swim. So, you will have some membrane protein which we have calculated. So, this is the membrane protein. Some ions it will have. So, these are inorganic ions. It will have some lipid molecules. It will have some protein. It will have its DNA. We can calculate actually how much the amount of DNA as well. It will have the ribosomes. And also it will have the messenger RNAs. It will have the messenger RNA. Okay. So, if you look at the numbers, we have already calculated, and water of course, we will have water which is H2O. So, if you look at the numbers, so water we said that it will have close to 2 times 10 to the power of 10. Membrane protein is somewhere on 10 to the power of 6. Ions are 6 times 10 to the power of 7. Lipids are 5 times 10 to the power of 7. Protein 2 times 10 to the power cytoplasmic protein. DNA will have 5 times 10 to the power of 6 base pair. Ribosomes we did not calculate, but you can do the similar calculation. Times messenger RNAs as well, you can do this. So, a complete E. coli constituents, one can do these kind of calculations to see how the entire E. coli cell will look like. Okay. So, you can see that the proteins, so typically are produced in one part of the cell, and they have to move across the other part of the cell, where this messenger RNA actually carry these protein from one part of the cell to another. Okay. So, what we have, this process is often referred to as a passive diffusion. So, there is a passive diffusion mechanism where these proteins are transferred or diffused from one cell to another, because the ribosomes, where again is the proteins are produced. So, through the messenger RNA that is done. Okay. So, if we do some calculations, you will see quite interesting fact. So, if I look at the passive diffusion process that is taking place in the cell. So, this is the diffusion of protein molecule from one end of the cell to another, which is the passive diffusion. So, at room temperature, the thermal energies we already know is in the order of kBT for any molecule. For any molecule, the thermal energy is kBT. So, if I plug in the numbers, it comes out to be 4 times 10 to the power of minus 21 joule. That means 4.1 piconewton meter. Piconewton nano times nanometer. Okay, important, nanometer. Piconewton times nanometer. Okay. Why I am interested in this kBT, the thermal energy of these molecules? Why we are interested in this thermal energy of the molecules at room temperature? In the context of passive diffusion of cell, where cell has to move protein from one end to another. Why this is critical?
It is somewhat related to the kinetic energy, and which is related to its movement. Diffusion is due to the thermal motion.
So, this is a diffusion is taking place, right? So, there are molecular motors within the cell. So, the molecular motors, so if you look at the distance traveled within the cell is around in the range of nanometer, okay, the molecules, and the molecular motors, their force is in the close to the piconewton. So, you can see the challenge is you have a thermal or kBT of the molecules are in the same order of which of the molecular motor energy. So, this creates a problem. So, your molecular motor, so molecular motors that are present, they will able to exert piconewton of force over a distance of nanometer. Hence, this becomes a comparable one over a distance of nanometer. So, your kBT energy becomes almost comparable to what you see in terms of the thermal forces. Say for example, protein with a 5 nanometer diameter, what will be the diffusion coefficient? How we can measure in water? What will be the diffusion coefficient of, because mostly is water, right? As I mentioned, 70% of it is water. So, protein has to diffuse with it through the water, right? So, what will be a how one can measure this diffusion coefficient of the protein in water? Is with the Stokes, or Einstein-Stokes equation, which is the kBT by the Draco 6 eta times the R. Okay. So, if I plug in the R, knew the viscosity of water, I know, correct, and kBT already I have calculated the thermal energy. So, that will give us the diffusion coefficient. So, if you look at, if you plug this number, the diffusion coefficient for protein in water is close to 100 micrometer square per second. Okay. So, what will be the time required for the protein to move across the E. coli cell? How much it will be? This will be in the microsecond, right? So, time scale time scale for diffusion over the E. coli cell, which is the entire length, we have already know what is the length of the E. coli is 1 micrometer. So, the time scale of the E. coli is L, length of the E. coli over the diffusion, length square over the diffusion. length square is 1 micrometer square over the diffusion coefficient, which is 100 micrometers per second. So micrometer cancels out, you get it close to 0.01 second. Okay? So it's extremely fast. So the protein will move within the E. coli cell at a very fast rate. Okay? Now think about the time scale. Say for example, if this was the key, the length scale for other things, like for example in our typically axon, right? So a typical in a neural system, in a nervous system, so typical axon length is close to 10 centimeter. Okay. So if it's such a large scale, so what will be a typical, what will be the diffusion time scale for these systems? If it is an axon that we have in our central nervous system, they are really long, right? So now if the protein has to move along an axon, what will be the time scale required for this passive diffusion? Huh? What? 10 centimeter? Yeah. So what will be? It will be in microsecond or what? Huh? In second? So any idea? Will be? So it will be a lot, right? So it is almost like 10 to the power of 8 second. So it's unfeasible. If a protein has to move in axon, is it possible? So passive diffusion cannot do that. So how one can move in these kind of large system? E. coli is fine, I can do a passive diffusion, but in our central brain and other places where there is a longer length scale of the problem, how then protein will diffuse? Huh? So you have to have some active diffusion mechanism. So what is an active process, diffusion process in the cell? So you need to provide some energy, right? So in a cell, which thing provides energy? ATP. So you have to have an ATP converted into ADP and the phosphate. So that's a cycle that will provide you with the energy. Okay. So of course these kind of things will not work. So you have to have an active diffusion process for large kind of system. So you need active diffusion. And typically the ATPs are, with the use of ATPs are consumed, which will provide these kind of system. So what happens is in nature of this, it is, as you can see, for a longer system it is not possible, right? So the cell needs to, in a certain location it needs to perforate, right? So that you create something called ion channel. So you have to create, over time it should create some sort of an ion channel, so to allow these kind of diffusion process to take place. So if you look at typically across an ion channel, because moment you create a pathway, then there is a easier process, because you have created permeability into the system, so the diffusion will take place much easier. So what happens is, if you look at the lipid bilayer, so if I consider this is the lipid bilayer, as there is there is not enough opportunity for the molecules to transport or diffuse, because there is the time scales are large. So there will be certain molecules, they will try to accumulate on one side of the lipid bilayer. Okay, so these are the various molecules, it will accumulate. So once you have accumulation of molecule, on the other side of the lipid bilayer there is nothing, right? So there will be an ion channel will be created. So there will be an ion channel, and then you can pump this using a concentration gradient. Okay, so the molecules can move in this direction using concentration gradient. You can also, through a ATP motor, you can also move things. So if there is an ATP motor running, you will able to pump a molecule across the concentration gradient. So this is your ion channel. So you can move the molecule across the concentration gradient using an ATP cycle. So ATP getting converted into ADP plus phosphates, inorganic phosphate. Pi is the inorganic phosphate. So the cellular mechanics are very complicated. So you can push through an ion channel through concentration gradient. At the same time, you can you can move things across the membrane using the ATP motors to move things across the concentration gradients. Questions here? So these understandings were critical for us to go inside understanding the diffusion process, the time scale. Well, the cell will perforate in order to have a proper sensing mechanism developed and so forth. So with these backgrounds, what we did is we look at the particular chemistry of the enzymes that are secreted from the E. coli. So the E. coli, the coliform, they produce two distinct kind of enzyme, which is the beta-galactosidase and beta-glucuronidase. Okay, so these are the indicator organisms that can actually help us to detect E. coli if there is present in water. So there are various kind of enzymatic substrates that are available in the market, or these are big molecular change as shown here. In this case, I picked two such enzymatic substrate, which is Red Gal and mug. So the one of the substrate interacts only particularly with the beta-galactosidase, and the other one with the mug, which is with the beta-glucuronidase. So the cell, they proliferate. So in other words, these ion channels are formed, so at a certain time instant, and then the enzymes are released, and then they react with these kind of substrates. Okay, so if I look at the reaction, how these molecules react, so if there is a Red Gal, so these are long chain molecule, you can see 6-chloro-3-iodiol beta galactosidase molecule, so it's a big chain. So when galactosidase, which is released from the E. coli cell, it cleaves this molecule, this big chain molecule, it cleaves it, and then it produces an indigo, which is a red color. Okay, so this reaction takes place because of the enzyme that is present in the E. coli. However, as in nature things are very slow, this will be extremely slow. It takes maybe days to do this reaction where you will see the red color. Similarly, the beta-glucuronidase, which reacts with a mug, which again cleaves it, but in this case you get a fluorogenic product. So in other words, if I excite this molecule with a 350 nanometer wavelength light source, I will get a blue emission. which is in the 460 nanometer. Okay, so this is a fluorogenic molecule which will emit blue color. And again, these reactions are slow. So the idea was to see how we can accelerate the entire process of enzymatic reaction as well as its reactivity with this substrate. So what we did is we put some lysing agent, which is a beeper. It is a lysing agent which allow to quickly lyse the cell, and then we put some oxidizing agent, which is the ferric chloride, which allows to have a rapid production of the color. And then we put red gall and mug in a broth solution, because that will keep the cell live, because this reaction needs the E. coli to remain live. If they die, then that enzymatic process will not happen. So there are these things we got to know as we work on this. So like any good chemist, we started in these kind of centrifuge tube, and we looked at if there was no E. coli versus if there were 4 cells of E. coli all the way to large amount of E. coli, the color of the intensity changes quite dramatically. Okay, but these works in a lab environment. Okay, centrifuge tube and so on. So we, as an engineer, we want to make a device out of it. So what we did is we made something called a field deployable mobile water kit. So what it does is the same, all these chemistry, understanding of the biological process and so forth, we have to create a product, right? Because people wants to test whether there is E. coli water. I cannot tell them there is ion channel forming and all these things. They said, okay, that's fine, but give me a solution. Okay, so we provided a solution. So what we did is we take water, which is a 100 ml syringe, and we put this syringe inside these filters. These filters have a particular cutoff pore, which is in this case 0.45 micron. So if you look at the initially I talked about E. coli, the length scale of E. coli is 1 micron. So in other words, if in this water, if there was an E. coli, then that E. coli will get trapped on the surface of that on that filter. Okay, and then we apply this A, B, C, D's are nothing. There are 4 compounds, which is the same compound that are shown here. So there is a LTB in mug, beeper, and then oxidizing agent and red gall. Okay. So the magic happens. So if there is an E. coli, you can see on the surface, because we have trapped this, kept it alive because of the broth solution, and then it lyses, enzymatic reaction takes place, and then there is an E. coli. And then we realize even though people don't have access to clean water, but everyone has access to cell phone. So we developed an app with Tata Consultancy Services. They are good in software, so I partnered with them to develop this app. So what it does is it goes to different wells. We went to different well, take an image, send that image to the server, and then it does an image comparison, because if there was no E. coli, then there will be a pristine surface. So it does a image pattern algorithm. So quickly says that, well, in this case it is sending, yes, there is E. coli present, but so what? So what we did is we collected all the cell phone or the mobile number of all the users of that given well, and we can send text message. So it is sending a text message saying that, well, 3 water sources found positive, you shouldn't be drinking water. Okay, so you can see how you can fuse technologies and create. For them, they don't understand anything of these nature. For them, that is very critical, because they want to know whether I can drink that water from the well or not. Okay. So what we did is, it needs a lot of work. So I worked with these are hospital in Mumbai. So they have a tertiary healthcare unit. So these are really very different kind of what I call it is a limited resource setting community. So it is completely things that you don't see it here. People actually drink water from these wells. So these are extremely poor neighborhoods in India. So this is around close to 40 kilometers outside Mumbai. So we take care of ASHA workers. So these are the people who are actually NGOs kind of thing, who will go and do the testing, because you need people to work and test with this to do regular testing. And then we also were sending water to a regular microbiology lab, because you have to make sure that whatever our mobile water kit was able to do, whether it was doing correctly or not, we have to do a cross validation with actually when you, in a normal case, what we'll do is you collect the water, send it into the lab like this kind of thing. So then they will grow, they culture the bacteria, grow it, and then they do a plate count. So the entire process takes 48 hours or close to a day or more than a day. By the time, if there is a contamination, people would have been drinking water. But our process is so fast, so that within an hour you get to know that whether there is an E. coli present or not through the color change, and then it avoids any kind of people drinking water, which is a big public health concern, because 70% of the diseases are due to waterborne diseases. So you can see that in these kind of neighborhood, you will amaze that the way, this is what they call it as a water purification system. So it is, what it is this is a river, which is a Kharagpur is the river. So in there the water is all collected into a big reservoir kind of thing, so an open system, and then from there is a pump which is going out. So every day this guy actually puts some chlorine, which has no idea what is, he just dumps it. And you can imagine, if you're an engineer, you can imagine this is completely bizarre, because it's an open system. You have flow coming in, flow coming out. So how you will able to control any kind of microbiology within this process or chemical process? So chlorination doesn't work, and that's why people were getting affected and so forth. So these are to my students and team members, organizations with whom we are working to do actual testing in the labs. So what we did is basically we do everything in the 96 well plate and so on, and then what I call it is a translational research, because we, at the end of the day, we are able to provide a solution to the to the community, and the community is worried, or they are more concerned about the solution. They don't know, they are least bothered about what is the front end in terms of doing things in the 96 well plates and things like that we did. Also, we did some miniaturization. So the entire thing that in a kit can be actually done in a wells. So these 96 well plates are actually a traditional way of doing microbiology or chemistry, but you can do these wells in a silicon wafer as well. So you can see we are able to do very fine wells which are 100 micron. So moment you make it such a small footprint, so surface area to volume increases, reaction rates are very fast, and we can do that very quickly as well, but it's an expensive process. So this cannot work in India. It can work in certain scale of economy, but not a silicon chip in India. It will never work if you have to do microfabrications. What we then realize is there a way to also provide a solution, ready solution for urban dwellers. So for example, at home you have something called a AquaGuard or some water purification system, but you faithfully kind of think that, well, this is working. There is no testing. You can't able to do any testing at home, whether that water that you are getting, whether there is UV has been cured or any malfunction of the lamp, and then you can get water accurate, reliably or not. So what we thought is, everyone has access to these kind of, in urban setting you can get this glucometer. So the glucometer measures glucose in the blood sample. So what it has is, it has a paper strip, is a paper microfluidics. If you put a blood sample which has a glucose through electrochemical reaction, then it will tell you what is the amount of glucose in the blood sample. We tweak the chemistry of the entire process, and then we, through the same process, E. coli, but in this we added some lactose molecule. So D-lactose molecule, when we add, the output of this becomes a glucose. So we tweak the chemistry, and so the output, if it is glucose, then you can use that in using a normal glucometer, and you can measure if there is an E. coli present in it. So that's what we did, is you now dip. So you take water out of your tap, which is in AquaGuard or something coming out, and then you dip the strip and add lactose molecule. The lactose is benign. There is essentially sugary molecule, and then you will get the reading if there is an E. coli present in or not. So you can provide various kind of solution based on the need in terms of whether it's an urban dweller versus people in the limited resource communities. I will quickly show you how we have further this is not in your presentation, how we have further modified this solution as well. So it is just hot out of the press. So we just got a cover article in Analyst. So what we did is we looked at so this is if you have to do a traditional testing. This is not coming. Something has happened. You do it one at a time. Okay. So this is a typically one. If you have to do it in a industrial setup or in a lab, you use this plate count. There is a coliform, E.coli, simplate, coli plate, and all these things are readily available in the market. But as I mentioned, it takes a huge amount of time. So we look at all this time. We said, well, let's not restrict only to Red Gal or mug. Let's look at all various kind of enzymatic substrate that can be used. So we have the various kind of enzymatic substrate. We looked at 21 different water samples. So it will have various E. coli, some other strains of microbes, and also we realized that a real water will not only have E. coli but also metal contaminants. So we want to understand whether our this system have any implication if there was some sodium fluoride, because water fluoride is always mixed in terms of municipality water. If there is some lead, cadmium, and so on. So we have just figure out, let's do all possible combinations that we see in a typical system. And then as a systematically, these are 96 well plate. So we did we looked at the various combination of these chemistries and so forth, and then we optimize this. So in the sense that if you have now we narrowed down only to 3 chemicals, so Red Gal, BPERD, and the LTB. That should be good enough in terms of whether the colors are produced. Other where there is no E. coli, there is no color, and even in presence of these metal ions and so forth, you will still see color is producing. So this is good in terms of well plate, and so the way we have done with centrifuge too. But again, you cannot just bring this to community because they will say, what is this 96 well plate and so forth. So you have to provide a product. So what we did is very interesting. So there is a simple thing that often you use in terms of sample preparation is a centrifuge, is called tube plunger assembly. So in a tube plunger, what you have is the tube, you can pour water in, and then the plunger has that filter. So the plunger has that 0.45 micron filter. So if you push the plunger in, and then before doing that, we have kept at the bottom a hydrogel. So the hydrogel is a porous matrix where actually you can cage that 3 molecules, the Red Gal, BPERD, and the LTB inside the hydrogel. So now you don't need to dispense any liquid chemicals, all are caged, and now you push the plunger in because it has a filter. So it will, if there is an E. coli, it will get compressed and get concentrated within the hydrogel, so that it allows the reaction, and the filtrate water will move out, and then you can, if there is a color change, you can see that with the color change happening. So it is really some sort of innovation, I'll say. So now we have the three chemicals, we mix it, put it into hydrogel. As I mentioned, this hydrogel is a porous matrix. So these are simple step, anyone can do it. You pour the water, plunge it, and then take the image of with the cell phone. So if there is an E. coli present, you will see that at the bottom, the hydrogel, there is a color change, which is the red versus, and this is small, so use and throw. So you can imagine is really an innovative way of solving this problem. So we can look at no E. coli to number of, as the color changes and so forth, we can, and this is essentially, this is how our cover article looks like. So what we are saying is that this hydrogel is a porous matrix which allow the materials where these chemicals can be added in, and they can be sent across anywhere in the world. So this plunger tube, we can send it anywhere with the hydrogel in it, and anyone can do this kind of test. So it becomes a technology for the masses. Any question on this before we move to the next topic? So the idea is, reason I'm showing all these things because at the end, what we do, it has to have some impact in terms of the society, and society in general is not really concerned about all these jumbo mumbo equations and things like that. They want solution, and that's where I think sometimes we get disconnected, because as a scientist we always trying to look at all these equations and things like that, but sometimes it doesn't work for the real life problems. Other things that we did is in the pathogens in food. So in India, for example, if you go to any of the supermarkets, you get this ready to eat meat products, so these salamis and so forth. But these are very dangerous in terms of, if there is some listeria, which is Listeria monocytogenes, is one of the foodborne pathogens, if they are present. So the way it come in is basically this is a meat processing line. So where the meat get processed, that line can get contaminated with listeria. So if that happens, they go the market, you get it, you are buying this and you eating it, and by the time the inspector gets in, takes a sample, already these meats have been packaged and it in the supermarket store, and people start consuming. And we had this incident in 2008, whereas the maple leaf is one of the famous packaged meat producer, and there was a huge amount of lawsuits and things like that. So it is a big challenge. So what we did is we used some high school physics to resolve, to find out a quick way to detect these kind of food pathogen. So if you recall, you have learned in high school physics is that if light is incident at a particular angle, it gets total internal reflection. Okay, so that depends upon the index of the material. So this is the waveguide with which the light pass, and the surrounding media, which is outside. Okay, when such internal reflection happen, there is a evanescent wave field which is close to 100 nanometer, very close to the surface at the interface, where the strength of that field exponentially decays. So the highest at the surface, if you go down up 100 nanometer, very sensitive, within the 100 nanometer is decay. So that is an evanescent field. So if I have a mechanism to distort that evanescent field, then it becomes a very sensitive biosensor. So that's what we did. We were able to make something which will able to distort this evanescent field. How we have done this is as follows. So we apply something in electrical engineering people have learned, which is called Mach-Zehnder interferometer. So what does this Mach-Zehnder interferometer does is as follows. So it has so the MZ so what you have is this is the waveguide. It branches out into two streams. Okay. So one is referred to as sensing, and the other one is referred to as a reference arm. And then they combine, and this is the output end, okay, where you see the output of the wave. So the reason we use Mach-Zehnder, it needs to have a particular angle. This theta is important, and it needs to have a particular material so that the refractive index of the waveguide versus the surrounding can create a total internal reflection. Okay. Once we have the total internal reflection, if you recall, there is this. If this is my sensing arm, if I am able to distort the evanescent wave, versus if there is a reference arm where there is no distortion of evanescent wave, then when they combine, then the output will create some interference, and that interference is what we will detect as a signature for this molecule, in this case the listeria. And we are able to do this because it is highly sensitive. So this is what we have done. Of course, this as I mentioned, these kind of system, any micro nanofabrication process is not a solution for limited resource communities. It is, it needs a scale of economy. So why it is expensive is, if you look at the structure, the waveguide that through which the wave propagates has a very unique structure. It has a ridge which is 4 micron width with a height of 4 nanometer. This is extremely difficult microfabrication or nanofabrication technique, because over a 4 micron then you have this 4 nanometers. So what we did, we took a silicon wafer. In this case we have all silicon oxide, we have a silicon nitride. So as I talked last time, how you can grow silicon nitride. Then we have a silicon dioxide on which we were able to do this lithography step. Okay. And the sensing area is where we do a biofunctionalization. So in other words, we attach molecules. As I mentioned, the best way to attach any surface interaction is the covalent bonding. So we covalently bond molecules so that the listeria can sit in this in a happy mode, so that we get a strong binding. The reason why we need this ridge structure is because when a wave propagates within a medium, there are various modes of propagation. There is a fundamental, first order, second order, third order, and so forth. In order to have a Mach-Zehnder interferometer where you get an evanescent wave properly, it has to be in the fundamental mode. So for the fundamental mode to happen, that's why we need this kind of a ridge structure to take place. Okay. So in a biosensor, selectivity is very critical. Like in a real estate business, location, location, location. So similarly in a biosensor, selectivity, selectivity, selectivity. Your biosensor has to be selective. In this case, our goal is to look at Listeria monocytogenesis and not E. coli. So if I put a mixed culture, it should only capture the Listeria and not the E. coli. And you can see that in here we are able to capture only the Listeria. This is SEM image, by the way. It's pretty difficult to do a SEM of this kind of bacteria. As I mentioned in the cold work also, these are pretty challenging when you have living organism in a SEM. It is because SEM is everything evaporates. Okay, because there's a vacuum, right? So the cell is made of water. So you can imagine what will happen to the cell. But we are able to. So there are techniques to do this. is something called freeze dry. You have to freeze dry the sample ahead of time to do this kind of work. We also do some multiplexing. So one other very good mechanism is, other day we are talking about this cantilever in terms of the elastocapillarity effect. So these cantilevers can be used for detection. So for example, if there is a particular binding event takes place in the arm of or the surface of the cantilever, then the cantilever gets surface stresses. So because of the surface stress, then it can bend, and that bending can be used as a way to detect molecules. Say in this case, we are looking at myoglobin and troponin. Do you know why these are important? Why these molecules are important? Myoglobin and troponin. Not infectious. These are cardiac markers. So if someone is going through something called myocardial infarction, which is in common term referred to as a heart attack, so the protein in the blood level like myoglobin, troponin, CKMB, their amount increases quite dramatically. So if you're able to have a sensor which will able to detect this level of biomolecule in the sensor, then it becomes a very effective tool. So this we use some sort of a capillary assisted process because we understand capillary flow in our group. So we use capillary flow to actually biofunctionalize each of the arms. So in this case, one arm is measuring myoglobin. So in other words, the antibody binding of the myoglobin versus the other arm, which is of troponin. So we can have specific arms which will detect. So what we call in biosensing is a multiplexing. So multiplexing is a very advantage. So you can have various in a single footprint, in a single this is a device, so to speak. So in a single device, you can have various organisms or target molecules which you can detect at the same time. No, video may not work. I don't think video works here. Oh, it's working. Oh, yeah. You can see that we are moving this cantilever inside the groove to allow the capillary forces to take place and do this biofunctionalization. Okay, so I think that's it I have for you guys. If you have any question on this part of the talk, questions? Yeah. Any questions that you want to? We have some time. We can talk about. No questions? Yeah. So we will see you at 6:30. I believe that's the time this certificates will be given to you guys. Okay. So please come here for the certificate part. So we'll see you at 6:30. And if anyone interested in going for grad schools and in my group, please feel free to email me. My email is mitra with s at yorku.ca, and my group is MNT Lab. If you do a Google search, you can always see the website MNT Lab. So please feel free to look at to the lab website, as well as email me if you have any queries, question regarding any of the topics matter that we have discussed in here. So if you have anything, please feel free to email me, as well as you can visit the lab website, which is MNT Lab. Okay, so make sure that I'm pretty good in email response, so I'll be able to respond to you guys. Thank you so much. It was a pleasure having. Okay, so this is that's what.